Oh, maybe that was a bad idea. Now I'm going to trip up that stuff. Okay, so what I'm going to talk about today is sort of two different components that are intertwined. First, I want to talk about a review we've been doing on phylogeography, and that involves Joey Shaw and Charlotte Germain-Aubrey. And then after that, I'm going to talk about this big grand idea I have for a collaborative network for collecting high-resolution data for phylogeographic data sets. And some of you have heard me talk about this before. So I'll just jump right into it. Phylogeography, by definition, is markers in space and time, and so you would think those would be the emphases of phylogeographic studies. And I've put some nice imagery here from a study that's in the review that we're doing, where you can see time and space and markers all included in there. And after 30 years of phylogeography, it was kind of my perception that animal studies far outpaced plant studies. Organelle sequences were most common, and the Pleistocene was the most commonly cited time scale for the events that were observed, or the breaks that were observed. And so those were my perceptions, but I wanted to evaluate that in a more empirical way, and that's when we set out to do a review. And I keep trying to click that. So you're probably saying, oh, do we really need another phylogeography review? Because you can find a lot of them out there. In fact, this was one that I was on several years ago. But most of the reviews that have been published have been with respect to pattern. What can we say in terms of comparative patterns? Can we detect common trends and things of that nature? And then, of course, Joey Shaw and his collaborators and I have done some papers related to chloroplast diversity and how we can use different components of the chloroplast genome more effectively for phylogeographic studies. But no review to date has addressed specifically markers in time and space in plant phylogeography, and that's what we set out to do here. So our big questions were, first of all, what markers are being used and why are they being used? Is it just because somebody else did it, or is there some legitimate logic to the selection of markers? Something I'm particularly interested in is divergence time estimation. So for all these studies that refer to the Pleistocene, are they actually timing these events in any empirical way, or are they just assuming that it's the Pleistocene that's defining these patterns? And then how often are ecological niche models involved? There was a full-day ecological niche model workshop on Sunday, and we learned all sorts of ways you can use modeling. But one of the ways that I think of it is in paleo distribution models as a way to test phylogeographic hypotheses. So I wanted to know how often those are included. And then, is it possible for us to come up with data standards for both data collection and data reporting? Because one of the things we learned through this review is that data reporting is all over the map in terms of what information is reported along with the data that are collected. And I'll elaborate on that in a minute. So here are the details of the review, but basically we reviewed papers between 2007 and 2014, which initially recovered about 10,000 papers, so 10,000 phylogeographic studies during that 8-year time period. We chose that time period specifically because there were a few studies that came out around, and you might be familiar with the Beck review that came out in 2008, and that was based on data through 2006. And so we kind of wanted to pick up where he left off, as well as 2007 would give us sort of a starting point to look at how the tortoise and hare series had impacted marker selection in studies since that time. So we couldn't very well read nearly 10,000 papers for this review, so we refined using ACE to try to identify plant studies. We tried a couple of different routes. I initially used cpDNA, but then of course you're potentially excluding nuclear studies. So this was what we settled on, and that got us down to 1,134 results. And then those were filtered by the top 10 journals and got us down to 536. And then we had to read every single paper to actually make sure that it met the expectations that we were looking for. And so we ended up excluding some additional ones based on these criteria here. So we ended up with 303 papers to review. Most of the information we were looking for was not available in the abstract and often not available in the paper itself. We'd often have to go to supplemental data online to recover the information we were looking for. So you've probably seen me talk about this for several years now because we've been trying to get all this information together. So here are the number of papers, the 303 divided by the top 10 journals. You get a sense of where our community is publishing the plant phylogeographic literature. And I should emphasize, you might have seen one of my exclusion criteria is no more than 5 species. So we were looking at single species studies or species complex studies. So you can see more than half, well, almost half the studies are in these two journals, Molecular Ecology and Journal of Biogeography, as you might expect. And then you can see where these other journals fall out here. Some you may have expected and some maybe not. So in terms of frequency of marker classes used by year, so on the y-axis you have proportion of publication in which a marker class was used. On the x-axis you have the year of publication, and then the color lines are indicated here. So you have organellar sequences that would include both mitochondrial and chloroplasts, because of course there are a few papers here and there that use mitochondrial data. Nuclear sequences are coded in red. Largely dominant data are coded in green, and then microsatellites in purple here. And so the pattern that you see is an increase in organellar sequence data over time, so that ultimately close to 80% of all studies by 2014 are using organellar sequence data, as you might have expected. And then interestingly, but maybe not surprisingly, we see a downward shift in dominant data, and then slight increases in nuclear and microsatellite data. I'm assuming those are because of new methods that have allowed us to more easily access those types of markers for our various systems. We have loads of data beyond this for these markers, but I don't have time to talk about it here, so you'll just have to look for our paper when it comes out. We then looked at chloroplast DNA sequence data in particular. So Joey and I actually documented every single individual chloroplast marker that was used in each of those 303 papers. And that was not an easy task, I should say, because of people using different terminology for the same marker types, for example. So here, what I'm showing you is these are, above this blue dotted line, these are the top 10 performing non-coding chloroplast regions according to the tortoise and hare 4 series. These two regions, which I don't know if y'all can y'all read that in the back, or is the text too small? The two most commonly used regions in our review. So TERN-H, PSBA, and TERN-LF are the two most commonly used regions. So in blue, on the left side of the figure, you have number of papers in which the region was used. In red, on the right side of the figure, you have the tortoise and hare 4 average potentially informative characters. That's averaged across all of the angiosperms in which they were reviewed. So what you see is almost an inverse relationship to potential informativeness and the frequency of use in studies. Now this is just a snapshot of this figure. We actually have this for over 100 regions of the chloroplast genome, and the trend pretty much holds true. So that's something to consider. Divergence time estimation, again, on the x-axis, or sorry, the y-axis, you have proportion of publications, on the y, year of publications, and then here we have these composite bars where in blue you have inferred divergence, meaning the time was just implied based on something someone had published. There was no empirical assessment done there. And then alternatively in red, you have folks that have used approximate molecular clocks, or you have in green folks that have used fossil calibrations. And what we see over time, at least, is you have an increase in the empirical approach to divergence time, which is great, but we're still in a situation where nearly half of all articles are inferring the timing of the events. And oftentimes, at least from some of the assessments I've done, the ones that are including divergence time estimates are finding dates much older than the Pleistocene, as many of you know. And then when we think about ecological niche modeling, this was the most shocking part, I thought. 92% of those 303 studies used no form of ecological niche modeling, so no paleo models. 8% of the studies did, and of those, you can see still more than half are not doing divergence time. So it's a very small proportion of the 303 papers that are actually doing all of these things. So why does it matter and what does it mean? And I like to think of it in terms of resolution, and I know this is a bird, but it was pretty. And so it showed very nicely the difference between a high resolution image versus a low resolution image and the detail you can see in the picture. And you can think of the same thing in terms of our phylogeographic inference. Low resolution is going to give us a poor picture or a fuzzy picture of what's actually going on out there. And so resolution is important, which is why I think we as a community should work together to develop what we consider data standards for both collection and reporting of data. So I'm working, I've talked to folks at iDigBio about developing a workshop to that end, and so I'm working on that now. Now, so challenges to collecting high resolution data. You guys may know James Beck. He spoke a few days ago. This was the title of his talk, and he kindly gave me some pictures he uses to show difficulties of field sampling. So this was actually from Croatia, and it was a minefield in one of his collecting sites. So that's probably not something we would encounter here in North America. But he also posted things like this where both road names in opposite directions of the same name. So those are some silly examples, but you guys can probably appreciate the challenges of trying to sample high resolution population level sampling in large areas for widespread taxa. I was just talking to Ross McCauley about this in his Australia work. So James has managed to try to deal with that through sampling through herbaria, if you saw his talk, and that's great, but he admits it's still a relatively inefficient way to approach things. So what I am proposing is a network source data collection strategy for phylogeographic research, where we use our classrooms to collect data towards a common end. Okay, it's not terribly different from what our herbarium digitization communities are doing right now, trying to give data to the same common end. But in this scenario, we would have some common question that we're interested in addressing, and we can use individual classrooms, individual graduate students, what have you, to generate small data sets that contribute to a much larger collaborative data set that would allow us to get the high resolution we need for less human resources, for less money and less time, potentially. So this is based in part on the ARIN network, if you're familiar with that, the Ecological Research and Education Network, but then also based on my experiences with CERNEC, iDigBio, and then I've worked with some folks on a collaborative approach to connecting students and citizen science to collections. So the idea would be to propose this as an NSF RCN UBE proposal. And I did that last year as an incubator proposal, and it was unsuccessful, so I'm working to revise that now. But I'm not going to go through the details right now of the aims, because there's a lot of text there, I know, and the objectives, but I want to finish while I still have like a minute left to allow you guys time to ask questions. So thank you.
Do you have time for a few questions?
Roland.
Yeah, actually, my question is about the use of the markers.
Yes.
So, are the two markers that are sort of overly used, are they all easily identifiable
Yes.
or something like that?
Absolutely.
Okay.
Yeah. What we're finding through the comparative plastomic work that we're doing in my lab in different unrelated taxa is that actually even with next generation sequencing of those plastome data, some of those top performers are not amplifying in our next gen data, and I know some of my colleagues are seeing the same thing in their data sets. So stuff we're going to address. Thank you.